from our previous class. And so what we're going to be doing is so I think it's gonna have to be a group of three and then groups of two. And so this is just bonus. I know we're far away from meter four, but you know, kind of getting this is going to work. So I'm going to pose you the slide, and more than the right answer is kind of getting you an idea of how some of the questions of meter four are going to be. I told you I cannot just show you a picture, some words. Okay, so you can look at your notes, but you will do this in there. Okay. So I don't know if you work with maybe. So if you want to work, Lauren. Sure. So. Okay, so I am going to again show you this picture, and the important thing is that you can support your answer. That's the most important thing. Turn these off so that you can see them then I'll turn it on. Anybody wants to get closer and look at the pictures? Yeah.
Just to be clear, they removed that meter, right?
That's what it's kind of tricky. So that's one of the things that you have, that's why I put figure 8. So you will know with figure 8, you will know what the key to that is. So this will help you figure out. Also just telling you these are roots, these are seedlings, so this is not like a shoot, these are roots. That's it. Anybody want to take stock there? And the bonus. And it's theirs, but you can
Okay. Wait, we have
no.
Okay.
So think about figure, look at the data in figure 8, see what is measuring. Based on what that is measuring, hopefully that will tell you something about how that relates.
Like, it just doesn't make sense.
It's just that first two sides, like,
I think I know what's happening, but if I switch up.
We'll give you 3 more minutes. No, that's a different class.
Oh,
I think that's for freshman bio.
okay.
I just didn't want to take it down because I think they have a lot this afternoon.
More production and ways.
Yeah, no.
he is very. Yeah. He's just right this easy. It's can't be that hard.
Okay, 1 more minute.
Stress writing.
Okay, so anybody wants to share your thoughts about how figure A and figure B are related?
So the figure A is showing the, I'm assuming it's concentration for the IA in the plant. So that would correlate to if you look at B, because you can see in B, the wild type, which is the WCL, which I'm assuming hasn't had anything done to it, seems like it's got much more set up for growth than the other one. Its roots are obviously way longer. And so its shoots do look a little bit taller if they're all lined up straight. I don't know if they all are, but. And then compared to the YUC, which is obviously mutated, I'm assuming it must have disabled something that would be lowering the oxygen available to what you're seeing in A. And then in B, you're also seeing it with like the root size is dramatically shortened. And so not, they seem a bit stunted. So
So basically what you can see here is what I told you in the previous class that the active form of oxygen is inositolic acid again. So you, in all of the papers, again, they won't explain to you what this is. They are assuming that you look at this and you can understand they are measuring oxygen. So there's more oxygen in the plant than the wild type, which since it hasn't been modified. And then this one is just a mutant that probably, it's impaired in some kind of synthesis of active forms of oxygen, so it has less nucleic acid. And then that's related to phenotypes in here. So remember, we didn't really discuss oxygen in a lot of detail because I told you oxygen is involved in everything. So it's also involved in wood growth. So in general, in cell elongation, plant growth. So maybe they don't really discuss anything particular about the shoots, but clearly something is preventing plants from developing normal roots. So one of the effects of oxygen is also root growth. Okay, so we briefly talked, I don't know if you remember, we talked a couple of weeks ago about shade avoidance syndrome and how plants that are kind of in forest, when they feel shade, it's because they can detect more far red light than red light. Because plants prefer to absorb in blue and red. So if you're in the shade, then plants have probably already absorbed all the good sources of light, and you're left with some of the far red light. And then we briefly talked about how plants try to avoid that shade. If they think that they are in the shade, they will develop some specific phenotypes to try to get some more light. So based on that, which of these two plants do you think will show a typical phenotype for shade avoidance?
The tall one because it will be dry to reach the light.
Yes, so this one, right? This one is totally full light. This one is trying to do everything, grow taller, expand so that it can reach light. Now we're going to start seeing how they can interact. We will call but 7 orphans. So I want you to think about it and name 3 of them that you think would be involved in shaping a plant, allowing a plant to move. Also, when you mention them, you just have to say a little bit of why. That's involved. So anybody wants to start? Okay, just-
Well, like the three I could think of off the top of my head that would probably have.
Let's do one.
Okay. Well, I was gonna say that I feel like Austin did it, but I was gonna say cytokines.
Why?
Well, they help in cell division, which I feel like if you're trying to augment your shape of, like, say, a leaf or just your phenotype in general, like cytokines would be pretty beneficial because you could increase the cell division rate going on in a certain area, which would help make up for time to get you into the sunlight. So, if you didn't have sunlight, I feel like you'd be wanting to do. So,
Okay. So maybe cytokines reduce cell division. Anybody else? Yes.
Option.
Okay, yeah. So obviously, most of the time it's the correct answer. Why else?
Responsible for active dominance, also used to promote pollen to oxygen probably.
Yes. So remember, auxin is important in cell growth, and that's the key thing, apical dominance. So if you're trying to grow tall, you don't want to branch out. You're trying to grow tall. So you probably get reduced branching when you are high. Any other hormones that you think might be important? Yes.
I think it's gibberellins that help with cell elongation.
Yes. So remember, okay, I remember. The picture of the cabbage plants spraying gibberellin and how they were much shorter because cell elongation is induced and internal elongation is induced. So gibberellin also will be induced because then you want the plant to grow taller. Any more hormones involved in cell elongation? Plant growth. Is one of the ones we haven't covered is important cell growth. What does it say?
Is that involved in bleaching?
So you think it would be
So it'd be involved in like spraying out like the
okay? Well, yes. So like more. So acetic acid in more because of increasing branching. Is that what you're?
Yeah.
Okay. Well, maybe I think it's more like a secondary role in this case for acetic acid, but there's a bunch of them. So the ones that I got are oxygen, gibberellin, racinasterol, and cytokine. Oxygen, apical dominance. So gibberellin. So here, these are comparisons which I really like this picture. So as you can see, these comparisons are showing a plant with normal light and a plant with a shade of oil syndrome. So here's a cartoon plant. Also remember, I'm going to talk about this. Gibberellin does cell elongation and also briefly talk about how a plant fence is going to die because it doesn't have enough light. You really use the algorithm early on because let's reproduce and survive the next generation. So gibberellin also is important in using this algorithm. We will talk about racinasteroids today, cell division and relocation. And cytokine is interesting to me. Have to read a lot of chain of evidence to figure this out. Actually decreases. So apparently, the cells grow and elongate, but you don't necessarily want them to be back. Because you want you want more than the cell plate to expand. You really want more cells, you just want them large. Not so much divided, but large. So clearly there's a lot of overlap in here. And then if that wasn't enough, I found this, which clearly says like everything really here. So they also involve ethylene and strigolactone is the one that reduces branching. So whenever we're talking about reducing branching, strigolactone. Okay, so let's talk about abscisic acid, so ADA. Also, so from secondary pathway. What you know about ADA? If we don't have this, is this gene producing more ADA? And what is it more involved in generation? Yes.
SPO1 would grade ADA, right?
Okay, why?
Because ADA is involved in like in its cascade tolerance.
Yeah. So, do you think these plants lack SPO1? Because, so, let's think about this. If these plants don't have SPO1, so do you think these plants have more ADA or less ADA?
I think less.
Less ADA, right? So, SPO1 is probably involved in ADA biosynthesis. Because once you delete it, the plants don't biosynthesize so much ADA. If it was involved in ADA catabolism, then that would mean that we have more ADA. That makes sense? So SPO in this case is involved in ADA biosynthesis. Genes, the name of the gene is the important part. The important part is to associate that ADA is involved in tolerance to this case. And that's part of what you're doing in the lab. So it's involved in ADA biosynthesis. These plants have less ADA. They are less likely to adapt to drought stress. So, abscisic acid was usually implicated in root abscission, and that's why they call it abscisic acid. That's what they decided. It promotes senescence. It promotes senescence, it also promotes fruit ripening. And for the first time, we're talking about seeds. So, it inhibits cell growth. So, by inhibiting cell growth, directly related to seeds, it helps seeds remain dormant. So if you have dormant seeds and you're preventing cell growth, means that in terms of seeds, you are promoting that the seeds stay dormant and not really germinate. You need a lot of cell growth and cell division to break dormancy and allow seeds to germinate. And they induce somatic closure. So it's induced under drought stress. It helps stomata being closed. Here you can see this picture. This stomach. There's no ADA, and 45 minutes after being exposed to ADA. Regardless of what's happening, if the ADA stomach are going to be closed, and not just closing the stomach, but it will also basically alter gene expression in a way that the plant will initiate many signaling pathways associated with surviving drought stress. So many adaptations. The easiest one you see is stop. So it is a drought marker. So in the future for your presentation, in any case, you read ADA or they were detecting ADA or ADA markers. Just like with indole acid, they are not going to tell you we are trying to test drought stress. It's like this is what we're trying to test. It's like imply that they are trying to measure something related to that. Also, if they are applying ADA, then they are trying to test again some kind of drought sophistication.
do the one on the left, since it's if I remember correctly, the brassinoid. So if you're not producing brassinoid, then your cell division and I think elongation of the cells would be less.
So basically, brassinosteroids are involved in cell division and elongation. So if you mutate the genes involved in biosynthesis, plants are going to be, well, there's not enough cell division, not enough elongation, so besides shorter, like underdeveloped digits, less cell division, cell differentiation. So many things are important here. One of the key things is photomorphogenesis, so that aspect that, have you ever left potatoes in the dark? For that, what happens to those potatoes? How do they look like after a while?
Parts for roots.
Yeah, so that's interesting. You think those are roots, but they are really shoots. They look like roots. This potato is very visible, that they are really shoots. But why don't you know they are shoots? Because they are pale, like roots. Why? Because there's no light. You take those shoots and you put them in the light, they will eventually turn green. But you usually sow your potatoes in the dark, right? So, racinosteroid is inherent in photomorphogenesis, so that turning on chlorophyll synthesis when plants are exposed to light. When a seedling germinates, it will initially start germinating, but what makes it green is that light activates chlorophyll. If you start germinating a seedling in the dark, it's going to be what's called etiolated, which is just pale. Racinosteroid is important for that. Another key thing, luminescence, stress response, and clearly a lot of overlap. So it's hard to tell exactly what it does because it's one of the secondary hormones that it pushes the other hormones into doing. So. What does the do? And based on what it does, can you tell me which of these two plants, left or right, are not producing steroids? Yes.
So stimulation is the signature hormone that allows microfungus to reach the plant. I believe you can see, but I'm assuming the one that has more density of factors that are more out is the one that is not lacking.
So it's not lacking, you means the one that has it?
Yeah.
So you think this one is the one that has the mass gene and is really new, right?
Yes.
Okay, so you actually answered the fourth question about microbiome. But one of the most important things for strigolactose was that it inhibits branching. Okay. The one that has more branches actually has less strigolactone. Strigolactone reduces branching. This one lacks strigolactone. It reduces branching and reduces root development of secondary roots and hair roots. So in this case, actually, the one with less branching is the wild type, and this one is the one that has more branching. You need to adapt one and you're going to have more branching and more root growth. Answer already. So which plant is here to adapt? So the important thing is we'll be talking about microbiology, and spironolactone is essential in allowing microbiology development. So you repair spironolactone, and there's no microbiology. Even if you leave it, everything else. How exactly that's important, we don't know. So here's a list of stuff, and one of the stuff is promotes symbiotic interaction. And it is the first yellow assignment for today was this. How are plants in pathways? Really very different from animals in pathways.
That's a lot of mobile.
Okay, well, yes, yes, so, but about the signaling pathway themselves. So let's talk a little bit about animal signaling pathways. You usually have, receptor in the plasma membrane, and it perceives something politically, whatever. Something is perceived, initiates a response inside of the cell. And usually what you have is a molecule that goes to the nucleus, usually a transcription factor. For the most part, transcription factors bind to DNA, and what do they do? Yeah. Yes, so for the most part in animal pathways, for the most part you're inducing transcription. Something is off, and until you get the signal, it binds and something is off. For plants, it's the opposite. Usually what you have is actually, instead of basically an inducer binding and initiated basically the response. In dance, what you have is off usually because a repressor is bound. So instead of something having to travel and induce it, something is already bound, and the signal represses the repressor, and then things can stop. According to the book, that is faster. I don't know how much faster you can be than a signaling pathway in animals, but apparently this allows a faster method because everything is ready to go. It's just removing the repressor rather than turning things on. So that allows these plants to react. They usually have repressors bound to them, and the pathway or the hormone helps eventually degrade that repressor. Allows plants to respond. Okay, so now we're going to see from the 7 hormones how they all seem to do this. Just like ethylene, in senescence, dilation, it induces. You apply GBRL, what is the doing you then? Yes.
Delayed.
It delays. What about J?
I believe that
Induces. So we have a bunch of hormones apparently all doing this. So they all do their own thing, but they can all induce senescence. What does that mean? That they contribute in different ways, but if you exogenously apply a hormone, you're overriding all of the control mechanisms in the plant. You're forcing it to do whatever you want it to do. So if you apply ethylene, acetic acid, racemic acid, just monic acid, or salicylic acid, they will all induce senescence. So ethylene is a triggering factor. Okay. So in many genes, basically apply ethylene and many things are initiated. However, one important thing about ethylene is remember how we talked about how ethylene can make fruits ripen? Same thing happens with senescence. If you apply ethylene to a tomato that is green and hasn't fully developed, it won't ripen. It has to be at a stage where it's ready to respond to ethylene before it ripens. If you have a cell that is just a leaf that is just growing, starting to grow, still developing, and you apply ethylene, it is not going to senesce. Why? Because the leaf is just not ready. So starting to understand that there is multiple factors and basically that's called competency for senescence. So you need to be ready to start that signal cascade that is going to allow you to sense. ABA is a little bit different. ABA can be an inducer in the sense that it can accelerate senescence more than ABA. But again, you don't really have more than, like in terms of roots, it will induce senescence more than ripening. And in terms of leaves, it will make them mature faster and eventually senesce. So they all contribute in different ways. And just one is acid salicylic acid, they are more like secondary, so the most important ones are ethylene and acid. As I said, the important thing is the competency for senescence, which means that if you have these leaves that are just maturing and you like whatever, any of these hormones, they will senesce until they are fully mature and ready to initiate. Now, which hormones delay senescence? Oxygen is always an answer, yes. We talk about oxygen before we want to delay ripening of tomatoes, for example. Any more hormones? Yes. Cytokine. Okay. So, actually, I really like this example of cytokine. So, I don't know how common it's here, but we're in fall, and so if you're walking around again, I don't know how common these little miners in here. Sometimes these brownie can have these green spots. Those green spots are called green islands because these are islands. You have this whole leaf senescing, but this part refuses to senesce. Many times what happens is in those green islands you can find different pathogens, and in this specific case, we're talking about leaf miner caterpillars. So these green islands, when scientists started studying them, they found they contain considerable large quantities of plant hormones that weren't present as high even in the leaves that were green. It was like a particularly high in your face amount of hormone, and we already said cytokine. So cytokine allows this. And so now when you think about it, if you have the larvae, the caterpillar of this leaf miner that is, a moth eventually, it survives there, it overwinters there. Basically, everything dies, but the caterpillar manages to stay here and it's mining that leaf and survive. And increasing the chances that it's going to survive, and overwinter. So, and by recently I mean years after this story. It was suggested that there was something in the excrement of these caterpillars that was directly associated with the phenomenon of the green islands. Basically, if you didn't eat the excrement of the caterpillars, then the green islands would be red. And eventually they figured it was probably an endosymbiotic association with gut bacteria from the caterpillar that was allowing the green islands. So now you have this massive interaction. You have the caterpillar surviving and probably something in the gut of these caterpillars. There was eventually deposited as, excrement. Was precisely what was allowing the formation of these green islands.
Can you give the caterpillar antibiotics?
Yes, so you can give the caterpillar antibiotics, right? And that's exactly what they did. They did. The insects, normal insects, and you can still see the green islands. When they fed the insects antibiotics, then the green islands. After a lot of sequencing and microbiome studies, they found that, well, one of the things is they showed that the green islands had a lot of cytokines. Even the leaves that were green, normally green, not senescent in the plant, had much less cytokine. So this was a lot of cytokine present. And the antibiotic to the insects just basically couldn't produce cytokine. And then after a lot of sequencing, they realized it was probably increased awful bacteria. And if you've ever heard about how they are trying to sterilize malaria, mosquitoes, and all of that, that actually sterilizes, prevents males from reproducing in many species. So it's this massive association related to gut bacteria, gut endosymbiote associated with an insect that produces cytokine in the plant that allows it to survive and of course allows the bacteria to survive because they're both doing that. So that's all related to cytokine. So hormones are the isonations, auxin, cytokinins, and gibberellins. Yes. Sorry?
It's more susceptible, so it's definitely reduces. To the extent.
Yeah, so it's more susceptible to editing. So this is the regular one. So what happens is both plants were sprayed with editing for days. Okay? So basically what happened is this is what you normally expect from plants, right? Fully developed, you spray ethylene, and ethylene is not only involved in leaf senescence, but also in leaf abscission. So losing the leaves in foliage. Now that many leaves are falling, how are oxygen ethylene and the oxygen in foliage? Yes, it's not like that.
Well, this oxen, I have that, well, it's like apple growth, it's stopping, like higher concentrations of oxen is stopping the leaves from growing. Is that what's happening? And then it's growing taller instead. And then,
Sorry, can you say that again?
so like when you have apical dominance, you're growing taller than wider, and that's due to having more oxygen in the lower leaves.
Okay.
Is that right? And that's what's making the leaves fall off.
So yes, related to oxygen, but not so much. So oxygen is important in everything. Right? So one thing is apical dominance, and as long as oxygen is abundant, it suppresses the branching pattern. Yes, but also oxygen is important in maintaining the plant like young in a way. So it is related to having less oxygen. So here you have oxygen is usually abundant, somewhat flowing into this zone, right? Like this zone here. And then as the leaf starts senescing, we know many hormones induce senescence. But also what happens is that oxygen that usually delays senescence stops being produced. So you have the thing that delays senescence that stops, and the thing that induce senescence are increased. So it's from both sides. So you have oxygen, there's oxygen being here. So that's one of the things. Oxygen delays senescence and it stops being produced. What happens to the ethylene? And what happens to the oxygen? Yes.
Is ethylene given off near the oxygen zone, which causes it to be digested and moved on?
Yes. So the key thing here is that two things are happening. Oxygen stops being produced, and then ethylene starts doing its thing. So oxygen stops flowing in this zone. Then there's more ethylene in return because senescence is starting, and so ethylene is abundant. And also ethylene is abundant, there's less oxygen, so senescence is induced. But the key is this abscission zone. It's a very important specific layer of cells that will react to that change in hormones. And so that lower oxygen, higher ethylene will induce very specific cell-degrading enzymes that are going to degrade this layer that I know it's hard to see, but it's just slightly different from all other cells. Those cells will start producing cell-degrading enzymes that will detach at the end the leaf from the rest of the plant. And it just basically cannot happen unless you have this type of cells, and these cells only react to changes in oxygen and ethylene. So I just have here the summary. So the abscission zone is morphologically and biochemically layer of cells that is very different. And it's digested again by the change of hormones, and then eventually they'll be shed from the plant. Ask any questions? Good. Okay. So that's it. So I will. I'll see you Friday. Also, if I